In our last lesson, we learned that when genes are paired off and split in meiosis, it means that different gametes from the same parent may be carrying different information. So when the egg and sperm meet in fertilization, there is a new genetic combination. It makes a new organism that has some genes from mom and some genes from dad, but the new organism is definitely going to be different from both parents. Humans have learned that there are mathematical patterns that are involved in this kind of recombination, and scientists call this study of inheritance genetics. These patterns are so predictable that if we understand the patterns, we can use those patterns to help us to predict the traits of our offspring. So today we're going to learn about the language, the vocabulary that we use to describe these patterns. There is a space on your notes for you to write in your essential question, and the essential question is, what is the vocabulary of genes? When we discussed meiosis, we were talking about chromosomes, and after fertilization, we can see that each individual gets one chromosome from mom and one chromosome from dad. On that chromosome are small sections of DNA that we refer to as genes. Genes are actually going to code for a protein, which is then going to give us the trait. But the information that I get from mom and the information I get from dad about a particular gene may not be the same. For instance, dad may give me a gene for dimples while mom gives me a gene for no dimples. So there are actually two forms of that trait. This is what we refer to as an allele. An allele is one form of the gene for a trait. And so that we can keep track of the different versions, we refer to these simply as a letter, which you are going to see as we start actually using this vocabulary. Most of our genes are shown in two forms. The first form is the dominant allele. This is a piece of DNA that, if it is there, is always going to show its trait. The second allele is called recessive. It is a piece of DNA that, even though it is present, may sometimes be turned off and we will not see its trait. The DNA to have dimples is a dominant allele. So I'm going to use a big D to show this. That is a capital letter to show that this is a dominant allele. No dimples is the recessive allele. I'm going to signify that using a lowercase d because it may be turned off if it is in the presence of the dominant allele. After fertilization, I have one allele from mom and one allele from dad, and my cell will use the information from both of these to determine what I am actually going to look like. The allele combination that we just talked about is referred to as a genotype. It is defined as the allele combination in the zygote. Remember that the zygote is the fertilized egg. So when we say I got a big D, a dominant allele from mom, and a little d, a recessive allele from dad, these are the two letters that we're talking about. There are three possible allele combinations. I could get big D, two big letters. I could get one big D and one little d, or I could get two little d's. Each combination has its own name. Two big letters is referred to as homozygous dominant. Homo means same, and zygous refers to the fact that this is in the zygote. Dominant tells us what kind of allele it is, and since these are both large letters, big letters, we call this homozygous dominant. Little d is also the same allele in the zygote, except those copies are recessive, little d. The last combination is called heterozygous. Hetero means mixed, and again, zygous means in the zygote. You'll notice that it is not followed by dominant or recessive because there is one of each. Genotype is not something that we can see by looking at an organism because it's actually in its DNA, and as you know, we can't see the DNA. But the trait that we can see is called the phenotype, and it depends on what the genotypes actually are. In the case of dimples, if a big D is present in the genotype, it will shut down the little d. So big D will result in a child that has dimples. There's no need to shut anything down here because there is no little d. But big D, little d, the heterozygous combination, is going to result in dimples because the little d has been shut down. Little d is the only place where we're going to see a child with no dimples because there is no big D to shut that down. The genotypes and phenotypes occur in patterns, and that pattern is easy to see if we can use a Punnett square. A Punnett square is a tool that we use to organize the genes and predict offspring. The Punnett square shows us the results of meiosis and fertilization. On the top, we're going to put the gametes of one parent. On the side are going to be the gametes of the other parent. And when these combine, we will get the combination in the zygote. So move to the second page of your notes and find example 1. This is the example that we're going to use to walk through how we use the Punnett square. If I want to predict the offspring from two particular parents, a Punnett square is very useful. There are 5 steps to using a Punnett square to solve a problem and make a prediction. The first thing I need to do is to make a key. To make a key, I need to know what the dominant and recessive alleles are. So you can see up here at the top is the place where the key is. Now I'm going to use dimples as my example again, and I know that dimples is a dominant allele. So big A is going to be dimples, and little a is going to be no dimples. That's my recessive allele. The second thing it tells me is to write the genotypes of the parents. Now remember that the genotypes is the 2-letter combination, and they have already done that for me. So if I look up here, I can see the parents. I can see one parent is big A, and one parent is little a. What happens if these two parents have children? I'm going to set up and solve a Punnett square that is going to be my prediction. I'm going to say, just for sake of argument, that this is the mom. Mom can only give one egg to each of her children. So here is one egg that has an A in it, and here is the other A in a different egg. That means this is dad. Dad is going to give little a in his sperm. So this is going to be sperm 1, and this is sperm 2. Now, mom is homozygous dominant, dad is homozygous recessive. So now I'm going to solve the Punnett square. If this egg were to meet this sperm during fertilization, then the combination in the zygote is going to be big A, little a. Now we usually put the big letter first because the big letter is dominant, and it is generally going to show us what the phenotype is going to be. If this big A and this little a get together, it is going to be a big A, little a combination. If this egg and this sperm get together, it's going to be big A, little a. And if this egg and this sperm get together, it's going to be a big A, little a. So you can see that there are 4 possible combinations. They all happen to be the same combination, but there are 4 combinations. Now the next thing that we're going to do is we're going to figure out what these letters actually mean. We're going to figure out how many different kinds of genotypes we have. So if I look here at my possible ratios of genotypes for the children, I can see in this square there are 0 big A, big A. None of the children, which is what this square represents, are possible offspring. None of them have the big A combination. However, big A, little a, I have 1, 2, 3, 4 of these. There are 4 out of 4 big A, little a. And the last possible combination is the little a, the homozygous recessive. And if I look in that square, there are 0 of those. Now, the phenotypes I need to know from my key. So I'm basically going to take my key and I'm going to transfer it down here. I am going to be looking to see how many of my offspring have dimples and how many of my offspring have no dimples. I'm going to need to look at my genotypes to find that information. Dimples is a dominant phenotype. That means that in order to get a dimpled child, I would have to have a big A present. So that means if I look at my genotypes, these are the two genotypes that have big A's in them. Those are the ones that would be children with dimples. So if I look here, I have none of these guys have dimples, but 4 of these guys do. So 4 out of 4 of my offspring are going to have dimples. The only one that would have no dimples would be a genotype that does not have a big A in it. That would be the homozygous recessive, and I don't have any of those. This means that all of my offspring are going to have dimples. So now I'd like you to try it on your own. We're going to use the same trait, so you're still going to be using dimples and no dimples, but you're going to be doing breeding pair number 2. So stop the video, work through it the best you can, and then turn the video back on and check your answers. So check your answers. How did you do? If you're having trouble with this, we're going to do 2 more examples, and you can always come to office hours at 11 and talk through the examples with me. So let's go to breeding pair number 3. This time we're going to breed plants. We have tall plants and we have dwarf plants. Look at the key, stop the video, work out the Punnett squares, and then check your answers. How'd you do? Let's try one more example. Let's try breeding pair number 4. This time our trait is going to be colors in certain types of chickens, which is a dominant black color and a recessive blue color. So stop the video, work out the example, and then come back and check your answers. So hopefully you're getting the hang of this. If you're still having trouble, make sure that you come in and get a little bit of extra help. Now go ahead to the 3rd page and continue with the practice. Good luck.